Society of Canada. I will be the host for this online event. With no end in sight, the pandemic has shut down many cancer research trials across the country, and many researchers are not able to work at full capacity. Research funds have been reallocated to COVID-19 studies, testing, and treatment, resulting in a reduction of funds for cancer research. This has resulted in stalled momentum for blood cancer research that will likely have a lasting impact on cancer care. The LLSC researcher super team, led by Dr. Florian Kuchenbauer, is a group of blood cancer researchers from across the country that have come together to help raise funds and advocate for the critical need to support blood cancer research in Canada. This team is a group of leading experts in their fields with studies that aim to prevent, detect, and treat blood cancers. Each year, more than 35,000 Canadians and communities across the country participate in the Light the Night to raise funds for research and critical programs and services that help support Canadians at every step of their blood cancer experience. The researcher super team is fighting for an opportunity to advance existing research and develop new therapies for Canadians affected by blood cancer. Today, I am very pleased to have our researcher super team with us to answer your questions. As you know, this is a special Ask a Researcher webcast. When you registered, we ask that you submit any questions that you may have about blood cancer research. Those questions have been organized, and our researcher super team is going to answer as many questions as possible as we can in the next hour. Should you think of any questions during the course of this conversation, please write in the chat box, and time permitting, we will answer them. There are 11 members of our researcher super team, and it's my pleasure to introduce everyone. First, Dr. Florian Kuchenbauer. Dr. Florian Kuchenbauer is leading the researcher super team. In September 2018, Florian started as a clinician scientist within the Leukemia BMT program of British Columbia, and as a senior scientist at the Terry Fox Laboratory in British Columbia Cancer Research Center. Next, Dr. Virja Banerjee is a clinician scientist at Cancer Care Manitoba and a senior scientist at Cancer Care Manitoba's Research Institute. Dr. Jason Berman is a pediatric hematologist, oncologist based in Ottawa, professor of pediatrics at the University of Ottawa, and CEO and scientific director of the CHIO Research Institute. Dr. Fabio Contu is an imaging scientist at Cancer Care Manitoba for the Genomics Center for Cancer Research and Diagnosis. Dr. Trang Wang is the head of research laboratory on hematopoiesis and leukemia at the Institute of Research in Immunology and Cancer, professor of pharmacology in the Faculty of Medicine at the University of Montreal, and she received the Murray Margaret Memorial Award of the Leukemia and Lymphoma Society in 2010 for her research on childhood acute lymphoblastic leukemia. Dr. Mani Larignani is an associate professor and Shrum Chair in Biological Mechanisms of Disease at Simon Fraser University. Dr. Gilles Robichaud is a professor at the Chemistry and Biochemistry Department at the Université de Moncton in Moncton, New Brunswick. Dr. Guy Sauvageau is a principal investigator in the Molecular Genetics of Stem Cells Research Unit at the Institute for Research in Immunology and Cancer, the hematologist at the Stem Cell Transplant Center, Department of Hematology at Maisonneuve-Rosemont Hospital, and professor in the Department of Medicine at the University of Montreal. And Dr. Schimmer is the Director of Research, a senior scientist and staff physician at the Princess Margaret Cancer Centre at the University Health Network in Toronto. Dr. Schimmer is also a professor in the Department of Medicine, Medical Biophysics, and Institute of Medical Sciences at the University of Toronto. Lastly, two members of the team were unable to join us today, but I'd like to introduce you to Dr. Rodney Decoder and Dr. Sabine Mai, who are pictured on the screen. Dr. Decoder is a professor of microbiology and immunology at Western University in London, Ontario, and he's also cross-appointed into the Department of Oncology. Dr. Sabine Mai is a professor at the University of Manitoba and Cancer Care Manitoba, and she's a Canada Research Chair Tier 1. Once again, thank you all for being with us today, and now we're going to get started with the questions. This webcast, the intent of this webcast is to be an informal conversation about anything and everything related to blood cancer, advances in research and treatment, and how COVID has impacted blood cancer research in Canada. I'm going to direct a question to an individual on the panel and then open it up to anyone else who would like to answer or add to the answer. So this is directed to the panelists. If you'd like to add to the answer, please raise your hand or use the raise your hand function, and you can obviously add your piece. In the interest of time, we're going to try and keep the discussion of each question to no more than 5 minutes each. So with that, Let's begin. So I'd like to start this conversation with talking about the current research funding landscape, and I'll direct this first question to Dr. Kuchenbauer. So in relation and related to why this super team was started, can you speak to how the pandemic has impacted blood cancer research across the country, and why did you feel that forming a researcher super team was important for this?
So thank you very much. This is an important question. Considering that I'm relatively new to Canada and the Canadian system, I have a good comparison to, for example, some European systems like the German system. And one of the things I realized here is that COVID dramatically impacted on research, and a lot of my colleagues told me that their grants, and I was also affected, have been reduced. And this is money where people sit on. You employ people with this kind of money, so it is important. And it is. And when I talk to patients about this, and because they ask me how's research, how's COVID doing, what's the impact of COVID onto research, I told them about all these issues, and people weren't really aware of this. And then they thought about it. It's like, well, it makes sense because everything goes into COVID research. So this was kind of the motivation for me to raise this topic and discuss it with the Leukemia Lymphoma Society, and they were super supportive, and that's how this whole researcher team got started a few months ago. Overall, I have to say, for example, in Germany, research funding right now, they do the opposite approach. So these private agencies that are mainly governmental funded and have some private donors as well, they actually try to support German researchers by making sure that their funds are kept up and not being reduced. So this is like the opposite approach, because we try to export knowledge as a good. So there are different ways of handling this, and I think everybody has to be aware of that.
Thank you very much. Does anyone else have anything they'd like to add to that? Maybe how we can, what we can do to address this issue, and how we can make the situation better.
Maybe we should move to Germany.
You have to be in quarantine.
Dr. Schimmer, you had your hand up.
I guess, around, why forming the super team, I can tell you that in Toronto, when the Light the Night event happens, there's always a large contingent of blood cancer researchers and physicians who participate. And it's obviously very unfortunate that, we can't be together physically this year. But I think, the super team is just one of the many examples by which I think the research community, has an opportunity to come together with the larger community in support of blood cancer research. And in terms, of the timing of COVID, look, I mean, there's not a good time for a pandemic, but the time with respect to blood cancer research was absolutely terrible. We've not seen this kind of advancement before in terms of our understanding about why blood cancer happens, why it occurs, why it progresses, and it's being translated now into new diagnostic tests and new therapies. And it's again, not there's a good time for a pandemic, but what a shame to be slowing down at this point, when we were making such progress. And so, it's also a testament that we've got to find a way to move forward, even despite a pandemic, to make sure that we're continuing to make important progress, because, our patients, their supporters, they rely on us.
Does anyone else have anything they'd like to add? Okay, so I'm going to have the next question directed to Dr. Manny, Dr. Larignani, you're here. So I'd like to ask the next question to you if you're ready to go.
Sure.
Sure, great. So in relation to that last question, we've had a few questions about the realities of getting funding, and what the success rate is, how much money is available, and in an average year, what percentage of your time is used to secure funding?
Oh boy, yeah, that's a really, really good question. A lot. Getting research funding is more competitive than ever. It's always been competitive, but Florian Kuchenbauer and I were actually working on a grant yesterday and chatting about this. These days, it's not unheard of to have a success rate of 1 in 10, or 1 in 7, or 1 in 8. So for every research grant that we need to get, we have to prepare something like 7 or 8, sometimes up to 10 research grants, and submit 10 different research grants to maybe get one to be able to have funding to do our work. So it's ultra competitive. That's the current climate. Many of the major funding agencies like CIHR, the rate is just over 10%. So, and I serve on panels all the time. I have served on more than 30 grant adjudication panels over the last 10 years. And this is what's been my observation. Like, we constantly have to turn down excellent grants. And you have to be at the top of the very, very outstanding to get funded. So that means all these excellent ideas go unfunded. And that's the problem with Research in Canada that the public needs to be made aware of. And it's true in leukemia and lymphoma. It's true across any disease that we are vastly underfunded. And many, it's painful to see all these excellent ideas, excellent research grants, and they have to keep applying and get rejected. And it also takes a mental toll on us as researchers and our trainees too, knowing that, we have to put so much time into preparing a grant, and right from the get-go that at the very, very best, you might have a 10% chance. And here you might be sitting on a cure, or you might be sitting on we were working on a new drug for leukemia lymphoma, for example. We solved the structure of a molecule that causes lymphoma and causes drug resistance in lymphoma, and we had this drug that we could show in the lab was working, very, very promising. And we were trying to get funding for this. And we had publications. We had excellent publications in the high peer-reviewed, very internationally reputable journals to back it up. And, for example, that particular grant, I had to submit 6 times to get funded. So that's, month after month that I'm writing and I'm tweaking it and I'm responding to reviewer negative comments. Probably because there was somebody ahead of me in the line that had to, had even a stronger case that had to get funding. So I'm not saying that the system is not fair. I'm just saying that it's very, very underfunded.
Does anyone else have anything they'd like to add? Because I mean, besides throwing a lot of money at this situation, we have to come up with obviously another explanation. So, Dr. Berman, you had your hand up.
Yeah, thanks. Can you hear me okay? I just want to mention how the entire funding pipeline has been affected, particularly by COVID, and wearing my research institute leadership hat. In order for grants to be competitive, as Manny's just mentioned, in these national and international competitions, you need to have preliminary data that's compelling, and much of that work is supported by local hospitals and foundations to allow researchers to get that data to do some of those really innovative and pivotal studies. And of course, those foundations and their funding sources have also been affected by COVID. So right along the entire continuum, funding is becoming more challenging, which means that really innovative ideas are not happening, because that really, you need sort of that spark and support for that kind of spark for that research that may lead to something really promising in the future. And so this is just to remind people that we have a competitive environment and we have funding affected all the way along the process, which has made it really challenging for researchers.
And then, so then this question is going to vary depending on how long you've been in research, but how long on average do you think on a yearly basis do you spend outside of your research or clinical duties actually writing grants and not doing research? Because that's obviously speaks to the issue with funding. Can anyone address that? Maybe Florian, can you speak to that?
I think Versha wanted to answer this.
Oh, sorry, I didn't. Sorry, Versha, yes.
All right, sorry. Yes, so I think when you're both a physician and a scientist, the balance that you have to keep to try and maintain both aspects of your life your academic life are very difficult. And I think the pandemic actually changed a lot of how we did things. So in my career description or my job description, I'm technically supposed to have 75% protected time for research. and my clinical duties should be actually 0.25. But my clinical duties are somewhere at 0.45 of my time, in addition to the 75% time that I am supposed to allocate to research. So I still try and maintain that 75%, but it also means that I'm overstretched. And during the pandemic, we're working very differently than we ever have, both for personal responsibilities and academic responsibilities. A lot of institutes actually had their labs shut down entirely from a physical perspective, with no one being able to come in and the research halted. The other thing is my research focuses on primary patient samples. And if patients are converted to a virtual format for medical appointments, then our traditional paths on acquiring samples are also altered, especially if you require fresh samples opposed to frozen samples that are donated by our patients for our research. So there was many things that were disrupted due to the pandemic, which made the ability for us to write our grants, to generate preliminary data, require actually more time and more innovation in order to apply for grants. But it also means that people may not be able to go into the competitions as readily at this point in time, and may require more time, even though they were so close pre-pandemic, which is why we require the supports of our patient population, but also the granting agencies to allow us to move forward with our research.
Paul, can I add to this issue? So the funding situation has been difficult for several years, and I must admit that in the past years, funding from the Leukemia Lymphoma Society of Canada has made it possible to keep the momentum in acute leukemia and lymphoma and blood cancers, thanks to the input from funding at the LLSC. And we were fortunate that in the past 5 years or so, the funding budget from LLSC has significantly increased to keep the momentum and to remind everyone about the fabulous discoveries that Aaron mentioned, Dr. Schimmer mentioned. We are at the best time for discoveries, finding new ways to diagnose and treat blood cancers. And so the pandemic came really at there's never a good time for pandemics, of course, but the timing now has made it really more difficult, and it's a pity. So hopefully we can keep the momentum going.
Thank you very much. So we'd like to move on to the next question, and this is, I'm going to direct this to Dr. Kontu. We've had a question about what are the research dollars actually used for, and people know that money goes out for funding, but don't know the specifics of what the money is actually used for. So if you could just give a few examples of where a grant that say the Leukemia and Lymphoma Society would offer, what that money would be used for over the course of a year.
Okay, sure. So let's start by saying that research is not cheap. It's very expensive. So once the LLSC collects the money and they grant the grants and award the grants to the PIs, so that's when we start. So let's say that the majority, most of the time, the majority of the grant goes into salaries. However, there are limitations for the percentage of the grant you can use for salaries. So depending on how complex the experiments are, you may end up having, for example, a person with a high education or a very skilled technician to perform those experiments. So in that case, the money used for the salaries will be on the higher end, while instead if it's very simple, sometimes students may end up doing those experiments, so the salaries are on the lower end. However, if the salaries can vary a lot, what doesn't vary are the supplies, the materials that we use. So that's what people usually don't see, that these materials are expensive. So to run a simple experiment, you have, for example, many washes. So for those washes, you need salts, and those salts come not cheap. Sometimes a salt can cost up to $400, $600. For example, in our case, we do many experiments with probes, paints for chromosomes, and those can cost up to $4,000, $5,000. And if you need to run multiple experiments, that cost adds up very quickly. Or antibodies that are, for example, $1,000, $1,500. So that's very expensive. Usually it takes about 40% of the budget just for the supplies. But then there are also the hidden costs, like, for example, the shippings. For example, we collaborate with clinicians from Montreal. So we need to allocate part of the budget for the shippings of the patient samples from Montreal to Winnipeg. And that takes some money. Also facilities. So if your project, for example, if your lab does not have all the equipments to run all the experiments, you need to rely on external companies to run some experiments. Then there is an additional cost, and also cost for the publications. They are not cheap. So there are multiple costs, and people don't see how expensive it can be.
Perfect. Thank you very much. And Dr. Obisho, I'm going to direct the next question to you. Is when it comes to research, what is the biggest misconception? And in terms of, I think this is getting towards the fact that discoveries take time, and it's not something you're going to see results overnight. And the follow-up to that is, are research funds allocated according to what blood cancers in this case are most common?
Yes, thank you. So I would have to say that the biggest misconception is I often had my parents asking me, well, how does research work? I mean, do you try a certain molecule, it doesn't work? And we usually call it, you know, the pass or fail or the shotgun approach. But the research actually, you know, for it to be effective and have the most effective interventions, we need to know what is going on, and to be able to block or suppress certain mechanisms that allow cancer cells to allow disease to progress. That's what we're trying to do. But what we're realizing now is that cancer is not a one person or multi-person disease. These diseases are personalized for every human being, and what we're realizing, and the whole cancer scientific community and the clinical therapies, is that we have to adjust our therapeutic approach on more an individualized platform. So we have to look at the certain people based on their genetic code, how they will actually respond to certain treatment. Are they more resistant, or are they more susceptible? And this is what we have to find out. And we're only noticing these certain genetic signatures or profiles now, and how people differ from each other, all depending on the treatments. There was one time, I remember it not too long ago, we're talking maybe 10 to 15 years ago, some cancer treatments and blood cancer treatments were effective at 35% success rates. And that was acceptable in the scientific community, and is really, you know, disheartening to know that we're at this day and age of technology and innovation, that we're only there and we're not capable of giving more. So when we prepare an experiment in the lab, it takes a whole team, and in no way am I a one-man band. My success is measured by my whole team. And we have to take every parameter and every variable into consideration. The cells have to be in perfect shape to test certain stimuli or signals or stress. The animal models, the mice, they need to be a certain age, fed a certain diet. And we often make jokes that the planets have to be aligned, but you know, it often happens like that. And everything has to mesh together at a certain time point, and we have to follow certain data collection points, either that the disease is at a certain stage, or that the cancer cells have grown to a certain point. So this takes enormous resources in terms of time, energy, effort, and the biggest one of all, the funds provided by the LLSC.
Yes, Florian, thank you for that, Gilles. Thank you.
And I would like to add one thing. I love how passionate everybody is about research, but you know, we are also creating knowledge, and we are so we are training new students. We are training future researchers, and this is something that is not even mentioned in the grant. I mean, we besides teaching what we all have to do, everybody who works in our lab, we pass on knowledge, and they will eventually pass it on, and hopefully pursue research with the same enthusiasm. I think these kind of things are not even evaluated in any grant or in when we explain how research works. So I think that's important to mention.
Thank you very much. And we're going to move on to the next question that we have, and this is going to be directed to Dr. Wang. How blood cancer research advanced cancer care? And I think this is getting to the fact that most people aren't aware that blood cancer research has laid the groundwork for many of the current treatment options. So would you be able to speak to that, please?
Can you hear me?
Yes.
Yes, great. So blood cancer research has made a huge impact in the cancer field itself. And I would like to mention just two examples. The first one is cancer immunotherapy. By transplanting, so in blood cancers, among the first cure was the transplantation of stem cells, blood stem cells, to patients. And in doing so, clinicians realized that there should be a compatibility between the donor and the host. However, we also discovered that there is, so now we managed to avoid this, and we have very successful stem cell transplantation, and we can cure acute myeloblastic leukemia with a stem cell transplantation. However, during this process, we discovered, the community, we discovered that the cells that we transplanted, there is also a reaction that we were not expecting, is the graft against leukemia. So the transplanted graft will reject the leukemia. And that was the beginning of the discovery of immunotherapy. And now the whole cancer research field has been influenced, or I should say the therapeutic approaches are being influenced by this discovery. It was made possible with transplantation of blood stem cells. So that's my first example. And you heard a video of this on the LLSC website last week. My second point is the discovery of cancer stem cells. It was also in acute leukemia that it was discovered, and it's now influencing the field of cancer research. And several of the scientists here can talk about their work on leukemic stem cells. It was discovered in Canada. And now we're really building new therapeutic approaches to target these leukemic stem cells. So these are the two examples I can give of how blood cancer research has made a wide, very important impact on the whole field of cancer research.
Thank you very much. Does anyone have anything else they'd like to add quickly to this, please? Yes, Dr. Banerjee.
Yes, I also wanted to add that the discovery of actionable targets within the hematologic malignancies or blood cancers also started the initiation of targeted therapy as I think another way that blood cancers have led in the molecular understanding and the genetic underpinning of diseases, but also the development of treatments.
Thank you. Dr. Berman, did you have something quickly you'd like to add to that?
Yeah, I just wanted to add that you've heard about some of the more recent advances in treatment in terms of immunotherapy, but really all the cancer treatments that are received today for blood cancers are because of research that happened in the past. And that timeline has been maybe longer than we would like. And one thing COVID has really shown us is that when people work together cooperatively as a team, we can really expedite what happens in the lab and bringing that to patients, because we've seen that in a much more shortened timeline than we've ever seen before. And the hope is that we can use that paradigm going forward to really advance treatment from the lab to the clinic.
Thank you. And Dr. Sauvageau, we've had a question, I think, that relates to this, about the causes of blood cancer, and whether you can you speak to the understanding of the causes of blood cancer and whether we'll ever be able to predict or / prevent them.
Well, there's more and more evidence that we all see cancer as a genetic disease, as an acquired genetic disease, but there's more and more evidence that in a lot of cases there is predisposition. And the new technology, which again is quite expensive but quite useful in identifying these predispositions. We had examples of P53 being mutated in the past as one of the first predisposition to cancer, RB, with one of famous Canadian scientists on RB predisposing to certain cancer. But now we realize in the blood system that a lot of patients, and it's tough to put a number, but it's certainly much higher than what we thought initially, that people have predisposition. So therefore, if we know they have predisposition and we know the genes, we could be in a position eventually to therapeutically act before or right before the disease develops. So that's one good example. There's one quick thing I want to say, which is not necessarily part of this question, and it's really our research staff. I mean, many of us have spent decades to train these individuals, and they've built research teams with us, and these people are extremely valuable. And with COVID, we realized how research is critical to save our lives. And we're all looking at us, we're all stuck at home or in our office at work, and because of this COVID, we can't do much right now. And of course, we're all waiting for scientists to come up with solutions. And I would draw a parallel with what we've been doing for decades, and now being in a position where we could lose these important individuals that have built this with us because of lacking funds to support them. So I think that's another issue that we should be discussing.
Thank you for that. Does anyone else have anything they'd like to speak to about either of those issues? No? Okay then.
Maybe Paul?
Yes, Trang.
Yeah, can I add a point on this, the training of our research staff and of the next generation of scientists? I think it's been mentioned, and I would like to stress this point, how critical it is. We all, the discoveries that we make in the lab is possible because of all these researchers that work with us and help us. Really, it's a teamwork that help bring us to the point where we are. And we are, right now, if research funding is getting tight because of the situation, we are at the risk of losing this highly exceptional and valuable collaborators that we have within our team. Having said this, there is the next generation of scientists that we are training in our research lab. So they come in, they are trained in research, and they will be the next generation to discover new ways to treat, new understanding of blood cancer. So we really need to keep the momentum going and really build the future of blood cancer research.
Thank you. Dr. Berman, did you have something you wanted to add quickly to that?
I was just going to add that, this next generation and training them, those individuals need to see this as a viable career so that they continue to do this really important work and recognize how it's making an impact. And all the comments that we've heard earlier today around funding and the challenges, particularly in the COVID model, really make that a pressure point. And so our job as mentors is to support and encourage the bright talent across Canada to continue to work in this field and stay in this country. You've already heard about how much the advancements and the leadership that Canadian scientists have had in blood cancers. We want to see that continue to the next generation. And with the support of LLS and your generous donors, that's going to be a reality.
It's kind of nice to see, sorry to interrupt you, but so for example, in our little panel here, I'm using models that have in the same lab that have been established by Guy Sauvageau, and I'm looking at this data. So it's a little bit like this kind of shows me that people can do things, and it motivates me to see that people are successful who are maybe 1 or 2 years older than me, like Guy, and so I really think it's a valid point that we have to create like a positive environment for the future researchers, for the future physicians to work on cancer and give them some hope that this is not a kind of a futile system.
Right. Thank you. And we'd like to move the line of questioning now towards treatment. We had some questions regarding this, and I'll ask Dr. Banerjee, how has molecular testing changed prognosis and treatment for blood cancers? And then how has continuous therapy versus time-limited therapy impacted the care of patients with blood cancers?
So I think one of the greatest advances I've seen both during my 20 years of training and now 10 years of clinical practice was a paradigm shift in therapy from sort of targeting the cancer cell in a non-targeted manner, but using therapies that worked based on the knowledge we had about the biology and the technology we had to discover. What we've learned more and more now is that with recent advances in technology and the cost of that technology decreasing, we're now able to identify molecular aspects of the different blood cancers. And those can be very specific to a type of blood cancer and also the individual, which allows us to risk profile them to determine whether therapy is going to benefit them or not, and then help direct the therapy at those molecular abnormalities, moving away from more broad types of treatment to some focused treatments, which allow for better quality of life, improved survival, as well as a change in the longevity and the survivorship of our patients. So that's the first question. The second question is, with these newer therapies comes a difference in the dynamics of how patients are treated. Because of the toxicities of previous treatments and their efficacy, we could use treatments that helped eradicate the cancer cells from the blood system for longer periods of time and achieve what we would consider a deep remission. Some treatments are more chronic in nature, and we cannot achieve a deep remission, but we can suppress those cancer clones, and therefore we're able to keep people on treatment for longer durations, but able to control their cancer. And this is also a paradigm shift in terms of how we think of cancer treatments, from something that was short and time limited to chronic cancer suppressive therapies, because not every patient may be strong enough to receive the most aggressive types of treatments. So we've now broadened our ability to treat patients who may previously not have been eligible for some of those aggressive types of therapies. And now we don't have to discriminate our therapies based on age or frailty, but can use the biology to our advantage to allow more people to be treated.
Thank you very much. Does anyone else have anything they'd like to add to that response? Are there any specific examples we can give for treatment options out there? Dr. Schimmer?
Well, I think just to build on that, you know, I think what you heard is that we're in a very exciting time with our ability to monitor patients and rationally decide what type of treatment and how many treatments. And because you think about what historically has been done, and you think about some of the treatment that perhaps some of the listeners or their friends or family have received, you know, they receive a set number of cycles or rounds of chemotherapy. And why is it that number? For example, 6. Well, some of that number 6 is a little bit arbitrary. And we now know that 6 is probably the right number for a certain number of people. But in reality is that there's some that could have gotten away with having less treatment. And unfortunately, probably some who actually required more than 6 rounds. It was just, we don't know who fits into which category. But our ability now to monitor very low levels of the tumor, low tumor burden states, and monitor it by detecting exquisitely low levels of that cancer cell DNA in the blood, sets us in a position where we can now more rationally decide on how many of those rounds or cycles does any individual person needs. Maybe it is 6, but for some individuals, we'll be able to tell them that their 3 or 4 cycles was enough, and others, unfortunately, that they'll need to continue something beyond that 6. And so we're in a very exciting point. And just to probably highlight, you know, that LLS has been a major contributor to the advancement in that area, funding various researchers around the globe working on this problem and getting us to the point we are now.
Yes, Dr. Brown?
Yeah, so I just would add to what Dr. Schimmer has said as well. You know, as a pediatric oncologist, this tailoring of therapy for children is equally important, and maybe even more so because of the risk of long-term side effects, because we know that we may be curing many children with cancer, particularly with blood cancers, that then end up having long-term chronic effects, risks of secondary cancers, impacts on their hearing, on their growth, on their kidneys, on their hearts. And so by being able to tailor therapy using molecularly targeted therapies, but also protective drugs, and really focusing to give patients the therapy that they need and not a one-size-fits-all approach, we can continue to cure a high number of children with blood cancers, but at less cost.
Thank you. And that actually leads perfectly into the next question, which I was going to ask you about. We had a question about how treating a pediatric patient is different from treating that of an adult, and what extra considerations have to be taken into account. But you obviously just addressed that. So I wonder if you can speak to maybe what are the latest advances in treatment that are tailored specifically toward pediatric patients rather than adults, or older patients.
So thanks for that question. You know, I think it's important to remember that children get different types of blood cancers than adults, and even when those blood cancers have the same name as an adult cancer, acute lymphoblastic leukemia or acute myeloid leukemia or non-Hodgkin's lymphoma, they can be quite different in children than adults. Children tolerate therapy sometimes better than adults do because they have young, healthy organs without some of the comorbidities that older patients might have. But as I mentioned earlier, they're at risk of long-term side effects, particularly if those organs haven't fully developed. So, if you treat patients with drugs that can affect their hearing around the same time that they're just learning how to talk, clearly this is going to impact their ability to have functional speech later in life. You've heard about immunotherapy, and actually in immunotherapy, Pediatrics has really led the way with regards to CAR T-cell therapy. So this is the ability for your own lymphocytes, which are an important cell in the immune system, to fight your cancer. And actually much of the seminal work that showed that CAR T-cells can function occurred in children with acute lymphoblastic leukemia, and now many others are working to try to find CAR T cells that will work in other disorders such as acute myeloid leukemia. So immunotherapy has been important in pediatrics as well. You also have heard from Dr. Savageau about cancer predisposition syndromes, and more we're recognizing that children may have an underlying susceptibility to develop blood cancers. And as we start learning more and more about that and identifying kids that have bone marrow failure syndromes where the bone marrow doesn't work quite properly, and then that evolves to leukemia, we can identify those children and do surveillance and potentially develop treatments that can improve their bone marrow function and prevent that leukemia from developing in the first place. And we're doing some of that work in our lab with funding from LLS.
Thank you very much. Does anyone have anything else that they'd like to add to that response? Yes, Dr. Banerjee.
I guess I'd like to just address the other end of the spectrum, because I think in the past, a lot of treatments really had age limits to them. And I think the development of newer therapies and the selection of patients based on their previous medical conditions and how their organs work are allowing us to add newer treatments to all patients with multiple different types of blood cancers as well, and in a safe manner that does allow them to have an improved quality of life and the ability to control their cancer. So I think, you know, there was a time period in which we had hard stops or hard age sets. And we also took into consideration some aspects of the disease that made patients more difficult to treat. But I want to provide patients at any age with multiple other medical problems hope that there's a lot more that we're learning and a lot more that we can do for them that we've ever been able to do.
Thank you very much. And I think I would like to, and this all builds onto this question to Dr. Schimmer here, and this can be addressed by anyone as we we go through the response. But what are the key areas of research focus these days? And if we're looking at near-term breakthroughs on the short-term horizon, or 5 to 10, 15, 20 years out, what are we looking at, or what could we be expecting in the research treatment field?
So I think there's a lot of stuff that's happening that's exciting. It's based on understanding the origins of cancer, why it happens, why it occurs, why it progresses, and then translation then into new clinical strategies, again, for the diagnosis or treatment. And, and at the end, we can come talk about some specifics. But one of the things which I would say at the higher level is why it's so important for investment in all facets of research. So clinical research that is testing new therapeutic strategies in the clinic, translational research that bridges the fundamental understandings of biology into new clinical trials, but also explain the importance of investing in more fundamental or basic research. Because it's through that greater understanding of why, again, blood cancers happen, why they occur, why they respond, and why sometimes they don't respond to treatment, that will then be leveraged into that next round of new therapies and new diagnostics. And so it's sometimes easy to understand why we're doing clinical research. It's sometimes a little harder to understand why it's important to invest in more fundamental research. But it's that fundamental research that pays its dividends in the years and in decades ahead. And if I could just bring it a little bit back to COVID, even, I would actually argue that the investments made in cancer and the blood cancer space have what's allowed us to be so rapid in our development around COVID diagnostics and ultimately therapeutics. You think nowadays, we can do a test for COVID that reads out in 15 minutes. Well, where did that come from? That actually came from investments that were made in cancer genomics and blood cancer genomics years ago. And in fact, had we not made those investments, we'd be back where we were in SARS, where we do a diagnostic test for COVID and the result comes back 2 weeks later. And imagine if we're back now in 1918 in the Spanish flu and we had not invested in science as a whole. where we would be. So in fact, the investments that we're making today in the fundamental understandings of blood cancer are going to pay their dividends in the years ahead, but not only for blood cancer, believe it or not only for cancers, but in many other areas beyond that. And of course, we as a panel can talk about some of the specific areas of advance, but I keep the comments a little bit high level with this response in terms of where some of the exciting things are.
Thank you very much. And yes, Dr. Ryan, you have something to add?
Yes. So I fully agree in the sense that we're bringing new knowledge into the field. And again, I would like to give one example how research in blood cancers have impacted on COVID-19 development of new vaccines, for example. So now we're learned to understand tumor-specific antigens, and it's much more unconventional than what we used to think. And again, I'd like to mention, you've heard about it last week, and I won't go through it again, but all this knowledge and this understanding in tumor-specific antigens is also feeding into the understanding of antigens in the SARS-CoV-2 to build better vaccines, to understand the antigenic epitope, to make better vaccines. So this is an example of how blood cancer research is very, very important to cure blood cancers, but also has impact into other diseases, including the current COVID-19.
Thank you very much. Now, we only have a few more minutes left here, so I'd like to ask some of the questions that have been coming up in the chat, and we can try to answer these fairly quickly because we only have 3 minutes. But I have a question here. COVID research for therapy and vaccines seems to be done in a more internationally cooperative way rather than the model of research labs competing in separate competitive silos. Is this an accurate view of the model of current research? Do you think there is hope for working more cooperatively in the future for the sake of patients? So, would anyone like to address that in a timely manner?
I can try this one. I think we frequently see confusion between, the mean and what is the essence. And by this, I mean, cooperation is a mean to achieve a goal. It shouldn't be the goal. And frequently I see this, and I would tend to disagree that there is a lack of collaboration, certainly not within, this group of scientists here. We all depend on other people's finding for making progress. And most of us work with, scientists, not just from Canada, but from all over the world. And it's really become a world community, an international community. We can't force collaboration where it shouldn't be, and we have to let the scientists find the right collaborators, and they do, because they know who's the best person for, the best program that they have. And it can be very, very specific and very specialized. So sometimes, your best collaborator is in New Zealand, sometimes he's in Italy, and so on and so forth. And frequently he's in Canada because we have a fantastic community of leukemia and blood scientists.
Does anyone else? Yes, Dr. Schimmer, last word to you.
Well, I would say, related to Guy's point, Guy is right. I think the biggest advances we've seen in science over the last while are the internet and FedEx. And the reason I'd say those is the internet allows, instant communication, and FedEx, which allows us to share reagents around the world. But I think there's also a little bit of what I would call friendly competition. And you need a bit of friendly competition because we don't know which is the right approach to solve any given problem. And by different groups taking different approaches is a way that you can actually achieve the goal fastest. And a little bit of competition moves things forward. But it's friendly because we also all recognize that when the solution has been found, there's no ego behind this. And when drops, what one is doing and moves, to embrace the solution, that's the one that's correct.
Thank you very much, and thank you everyone for all the answers, your insightful answers to the questions. We have had a number of questions come in, but unfortunately we don't have enough time to address all of them today. If you do have questions, please reach out to LLSC, and we will do our best to answer what you have. Dr. Kuchenbauer, do you have anything you would like to say very briefly, just to before I close out this this webinar?
No, I really hope we could spark some hope in terms of that. What we do is not too specific. Like, how should I phrase it? What we do is important, that we not only invest into our own careers, we invest into patients, and we invest into the future of research, like younger researchers. That's actually what's lasting. And I kind of want to bring this across. It's really important to see that it's not only about getting the right experiment done, it's way more.
Thank you very much. And with that, we've come to the end of the time that we have for the webcast. I want to thank everyone who is listening in, and would like to say a very big thank you to each member of the researcher super team for taking the time out of your busy schedules to answer these questions and to be with us today. 2020 has been an unprecedented year, to say the least. We know that cancer doesn't stop, and that the need to fundraise has never been greater, and that people want to help. Although Light the Night has had tremendous success over the last 15 years, we need to adjust our strategy to the current reality. So we are proud to announce that for the first time in our history, we will meet as one community on October 24th, 2020. From coast to coast, our community will come alive to share survivor stories, honor our heroes, and remember loved ones. Mark your calendars. The virtual Light the Night broadcast airs at 7 p.m. Eastern Time. Visit lightthenight.ca for more information or to register. And please remember to show your support for not only the researchers on this panel, but for every researcher across the country. Visit lightthenight.ca/researcher team, and visit the Light the Night Researcher Super Team page to show your support. Thank you all for joining us. Have a wonderful day and stay safe.
Thank you.
Thank you.
Bye now. Thank you for listening in.
Bye-bye.